Welcome, everybody. Thanks for coming to Grand Rounds. For those of you who are at the research forum that Mike Koob and I gave a few months ago now, there'll be an awful lot of overlap. So
recording in progress.
Check your messages and things like that. I'm afraid the same jokes as well. But there's some updates about our progress on this project. I'm gonna have to move this out of the way. There we go. So this was a project that a group of us, several people in lab medicine pathology, started last year to generate a new mouse model for studies on SARS-CoV-2 infection. And modeling the 99% is in part, I guess, a tribute to the anniversary of the Occupy Wall Street, but there's a more scientific reason for that title as well. The basic focus of the work was to generate mice that would have some of the key receptors that are involved in infection with SARS-CoV-2. That virus doesn't infect mouse cells typically. There is an important molecule called ACE2, angiotensin converting enzyme 2, expressed on cell surface of the host cells. That's important for attachment, and a protease called TMPRSS2 that's involved in activating the spike protein to allow it to attack ACE2 and enter the cell. Mike is going to talk about the details of generating the mouse models, but I'm going to give a little background for the context of how we got into this work, and then follow up with some of the experiments we've done on function. So at the outbreak of the pandemic, it was found that available mouse models were limited for studies on SARS-CoV-2. So normal mice are not infected by the original isolates of SARS-CoV-2. Some of the variants, of SARS-CoV-2 may be infectable, but the original ones that were studied were not capable of infecting mouse tissues. There were mice available developed by Stan Perlman at the University of Iowa that carried the human ACE2 expressed under promoter of K18 K18, the human keratin 18 promoter, giving strong expression in mouse lung epithelium, but also other tissues. It was found that the original virus SARS-CoV-1, as we should call it now, can infect those transgenic mice. They, as shown here in this graph, this is with SARS-CoV-2, but basically you see the same thing with the original SARS virus. You get a rapidly fatal disease infecting these K18 transgenic mice. And in large part, that fatality is due to infection in the brain. So it's shown here. If you measure viral loads in various tissues after infection of control mice, these are just the regular B6 background mice. They don't get infected with SARS-CoV-2 or SARS-CoV-1. But the K18 mice are infected. You get a pretty low viral load in the nasal turbinates. In the lung, there's pretty good infection initially, and then it kind of declines over time, as might be expected. But the viral load in the brain shoots up by many, many orders of magnitude. So useful to be able to have a model where the mice could be infected, and that allows us to do lots of experiments on things that will affect the infection and better define the pathology involved. But this infection in the brain was concerning because it's not especially typical, as far as we know, in most infections in humans. So these K18 mice are useful to study fatal COVID-19, but this is actually a rare consequence of SARS-CoV-2 infection in humans. Obviously, extremely important consequence, but we did want to be able to model what happens in non-lethal infections in order to look at longer-term effects of the infection, things such as long hauler syndrome and infections affecting other parts of the body apart from the lung. There we go. And if you kind of dig down into that in terms of what sort of percentages are we talking about of people go on to fatal infection with SARS-CoV-2, it's hovering around 1%, and that's part of the reason for the title. That about 99% of people who are infected don't go on to die from the infection. That's obviously partly a tribute to very efficient healthcare. But how do we get a better handle on that, the effects of that disease and long-term consequences? So last year, we proposed to generate and analyze mouse models, in actual fact, in which there's physiologically relevant expression and tissue distribution of human ACE2 and the human TMPRSS2 protease to model the non-lethal SARS-CoV-2 infection and subsequent immune responses. And this was a collaboration between several people, Mike, myself, Chris Hoquist, also in lab medicine pathology, and Tyler Bold in the Department of Medicine, who's an infectious disease guy. We responded to a notice of special interest, NOSI proposal for an emergency competitive revision. Don't go into the details, but essentially this was added as an administrative supplement to one of my grants on an entirely different topic. But that's the way they were supporting these investigations. So we submitted in April last year. It was awarded in June with a little delay in the start date. This has now expired, so we went through a no-cost extension and went through that. And at the moment now we're continuing this work with support from, in part from the generous support of the department through my professorship and through collaborations and various sources of funds to keep the work moving forward. Despite reduced funding, more people are involved because we're trying to get more comprehensive analysis done with limited numbers of animals that were infected. So I've listed the same people here and the people in the labs who have been doing the work. We've also enlisted the help of Jeff Hart and Maxim Chirin and folks in their labs. And I'm kind of acknowledging the people who provided viral stocks and made the chimeric mice and so forth. So it's a cast of thousands at the moment, and it'll be interesting when we put the paper together to see who's in what order. So the proposal was to use gene replacement approaches, which Mike will detail in a moment, to replace the mouse ACE2 gene locus with the human ACE2 locus, and likewise for the TMPRSS2 loci. Both of these mouse models, part of the I think the appeal of the proposal was that we weren't going to make these mice and then sit on them and work on them for 10 years and then finally give them away reluctantly to other people. The plan was that as soon as these mice were generated and initially characterized, they'd be sent for immediate distribution from Jackson Labs. And that is what we've done with the original ACE2 transgenic, gene replacement mice. There. Then we wanted to characterize SARS-CoV-2 viral growth, pathogenicity, and basic elements of the immune response in these mice. And with a special focus on the ACE2 gene replacement and the double gene replacement mice. And that's just to give an initial overview of the capability of these animals to be infected with the virus and compare this to the K18 human ACE2 mice, which at the time we sent them the proposal was one of the very few alternative options for looking at mouse models of SARS-CoV-2 infection. And obviously a fundamental question would we actually get infection? Does it lead to less severe disease and or viral spread to different tissues compared to the K18 model? What types of immune responses are induced by infection? And can this model be used to mimic the range of human COVID-19 disease? And obviously that's the biggie that we're still working towards, but we believe this will be a useful model. On to Mike, I think. So I'm going to stop here. Take over.
All right. Setting the scene. So February 2020, we were 5 months into a new project to model Alzheimer's disease-related dementias. So the goal of this project was to mimic the genetics of the human disease of dementia as closely as possible in the mouse. So to do that, it was based on new technology that we've been developing. And so to move the human genetics into the mouse, what I really mean is that we had to move the human genes into the mice. So what is a gene? There's a broad range of understanding of what a gene is, but most people agree it's a segment of DNA and it makes a protein. It's sort of the standard idea. And that's actually functionally the limit of many people's understanding of what a gene is. So I just looked on the internet and found a diagram of a gene, and it says it has a promoter that dictates how much the protein is made, the pattern of expression. There's a coding sequence, which they actually call the gene here, and then a poly A adenylation sequence, and that was it. So this is a gene in a very limited sense, but really this is a synthetic pseudogene. And I looked and just got a estimate of one of these types of genes that we got from someone else. The promoter, was a little over kb.5. The cDNA was a kb.5, and then a little bit of sequence for poly A. So this whole transgene is about 3.5 kb. So the reason people use these synthetic pseudogenes over time really is just because of the size. You can work with 3.5 kb, and then working with a real gene is actually very difficult because of the size. So how do you make a model from these transgenes? The standard approach has been really just to take that DNA, that transgene sequence, linearize it or not, and then shoot it into the pronuclei of an embryo. You get random integration at unknown sites, and you get random configuration of the transgene going in. So unfortunately, or fortunately, depending on what you're trying to do, no matter how many times you do this, you end up with a different result. The transgene will go in one place or a different place, and depending on where it goes in, your pattern of expression is different. So what do you do among all these tens or dozens of lines that you get? Really is just sort of look at the phenotype. So the phenotype is really dictating which model is selected from your transgene construct. And unfortunately or not, some people really just pick the phenotype that they liked. So you made a model for a particular reason. You looked to see what the phenotype was of the models that you got. You said, okay, this one works. All these others really didn't work. And so anyway, what does that mean functionally? So I published a paper just a few years ago where we looked in detail about what was really the molecular basis of phenotypes in one of those widely used models of dementia at the time. So the transgene again, and this is a transgene picture, again, just a small 1 kb of coding sequence, some untranslated regions on either side, a partial promoter here, a polyadenylation sequence, and then a synthetic promoter to drive expression. What we found wasn't just a single integration, but actually that the transgene had gone in over 70 times in an array. I'll move my picture here a little bit. And that configuration was really unusual. It just a long repeat of transcript one after the other, and then that shifted somewhere in the middle. I'm gonna move my picture a little bit here. Hold on. All right. Okay. And that the expression in this gene, in this line, was actually driven by another transgene. In that case, there weren't as many copies. There were 7 copies of the TET transactivator, but that replaced even a larger region of the mouse genome. In the original, or in the tau transgene, about 200 kb of genomic sequence was replaced, and that knocked out 3 out of the 4 isoforms of one gene, FGF14. In the case of the transgene driver cassette, that had gone in 7 times. It knocked out a half megabase of genomic sequence, and 5 genes in the genome were knocked out. So why was there this distinct phenotype in this line that made it really popular? Basically, it was because of all of the artifacts that came out from the random generation of the model. So there's massive overexpression of the transgene product. Over 700,000 bases of genomic DNA were deleted, including 6 genes. The TET transactivator itself was toxic and led to cell death. And then the pattern of the cell loss that made it look somewhat like human dementia was really dictated by a random integration events. This transactivator had a promoter, but it wasn't really the main driver of the pattern of expression, but really it was this integration event. And that region of the genome was dictating the pattern of expression. So how to do this in a more genetic approach that wasn't really quite so random. So our basis for generating models for dementia evolved to really trying to work with full genes. So full gene expression in humans and other mammals is very complicated and complex, but it's not random. So this is that same gene that encoded the KB of open reading frame that we used in the other model. But in this case, it's 190,000 bases, not 3.5 KB long. So the sense gene is here, but there's actually an anti-sense regulatory gene associated with that. And the regulatory region is here, over 50 KB of sequence with that. The expression isn't the same. We don't always make the same product from this gene. There's changes throughout our lives as we develop. The response of the gene changes with conditions. There's spatial regulation of gene expression is very cell-specific. Some cells make one form of the protein, others make a different form, and some don't make any at all. And there's lots of different products. With the cDNA, you get one product. With a full gene, you get a lot of RNA products and a lot of different protein products. So this non-random but complicated expression pattern is dictated by the genomic sequence, in this case, 190,000 bases of the human genome sequence. Anyway, so the complex gene expression really is dictated by the human genomic sequence and its placement in the genome. So our approach then, which was technically difficult, but we finally worked out the details of how to do it, was actually to take the full human gene and put it in place of the mouse version of that gene. All right. I am now not, there we go. All right, so that was February 2020, five months into this project. March of 2020, everything came to a halt. So the COVID pandemic restrictions took place. We were told to pause our research and wait for further notice. So rather than do that, I started to think, what can we do really to help with this problem? And obviously then we just decided that we would make a mouse model. And how do we do that? Our goals then were actually exactly the same as what the goals were for the dementia modeling project. And that's to mimic the genetics of this human disease as closely as possible in the mouse. So how does the mouse differ from the human in terms of its genetic components of SARS-CoV-2 infection? And the main components then are a receptor, the ACE2 receptor here, and a activator, a protease on the cell surface, which cleaves the spike protein and makes it active for binding and infection. So those are the two key targets then for generating a mouse model for SARS infection. So our goals at the beginning of this project then is replace the mouse ACE2 gene, which doesn't support SARS infection, with the full human gene. The mouse gene is 75 kb long in the mouse genome, with the human equivalent being 65 kb. So that was that target. The other then is a modifier of infection. The mouse TMPRSS2 gene is functional. We're able to get infection with the mouse gene, but our feeling was that we would get a more accurate modeling of human disease if we used the human modifier as well. These will all be on the C57 Black 6 background, just because to make it uniform. And the goal then was to make all of these gene products from human genes, express them in the pattern typical for the human gene. All right, so looking at the human genome, you see some flanking genes, the CLTRN, the BMX gene on the other side. That pattern is sort of maintained in the mouse, so you can kind of see really where this human gene should go. So that's how I designed it. I took the sequence from the end of this gene to the end of that gene, and then we did our crossover there and moved it in. There's a lot of details, but I find that people really aren't that interested in that. But functionally, this is what we have. And have. So in the end of this, we've made a ACE2 gene replacement model. 65 kb of human sequence, the full coding region, the whole regulatory region, and it's in precisely the same context within the mouse. We were able to do a full genome sequence of this allele and everything lined up. We didn't have any errors. So within this ACE2 region, this gene is indistinguishable from what we would find from the human donor of that gene. So this is all human sequence and then mouse on the other side of that. But does it work? Yes, it does, I guess is the bottom line. So with the RNA analysis, we have human transcription assays. You can see that the mouse ACE2 is not detected, but we see strong expression in the kidney and in the colon. And as illustration of the fact that this is not a cDNA but a full gene, you can see that there's actually two different forms of this region of the gene that we're assaying that you can pick up in the colon sample. On the protein level, it took a little hunting, but we were able to find a human-specific antibody. And with Western, the mouse does not make human ACE2 protein, but our ACE2 gene replacement line has a very nice signal for expression. We worked with the histology group and did some tissue staining. And the pattern of this expression is exactly what you would expect and what you do see for ACE2 expression in human tissue. This is lung. The particular cell types are the same that you see in human. Kidney has a very high expression. This is the angiotensin converting enzyme, and really a lot of the function is in the kidney, and that's where you see really very high levels of expression. Other tissues which do have a disease relevance include the heart, and again, we see appropriate levels of expression there. All right, so we've got the line. It contains all the potential therapeutic targets that you might use for therapies for COVID. That includes the full genomic DNA sequence, all the RNA transcription variants that it makes, and then all the protein products that it makes in the right levels. So this is not overexpression. It's just basically exactly what you would see from a human patient. The pattern's the same. The dynamic regulation should be the same. And these mice are devoid then of the mouse ACE2 protein when they're homozygous for this allele. So 6 months after the funding began, we were able to transfer these mice to Jackson Laboratories by December of last year. And then as of August, they distributed them. So these have been distributed. Experiments are being done on these in other labs around the world. This was non-restrictive. It's not particularly a collaboration with anyone. We're just sharing them. All right, so that part of the project worked out well. So replacing the mouse TMPRSS2 gene was very similar. We crossed the appropriate region. We were able to make the cross. We got the ESLs, the genomic sequence confirmed that it was correct, but we didn't get germline transmission. So in doing this as quickly as possible, we skipped a few quality control steps, and it turns out that the ESLs that we had generated had other chromosomal abnormalities. So we've since done this again. We've got this in, but again, it's a modifier of infection. So we're finishing this up without funding. All of our characterization work will be done with the TMPRSS2. All right, so I can stop there for now and transfer this back to Steve.
Thanks, Mike.
Yep.
So in order to go forward with the ACE2 gene replacement mice, we wanted to determine how intranasal infection with SARS-CoV-2, going from the original, widely used anyway, strain that was the Washington isolate. We wanted to check that actually we would get infection of this model and compare it to the other strains that I've mentioned, the K18 transgenic mice and obviously wild type non-gene replacement or transgenic controls. So we started with an intranasal infection with SARS-CoV-2, and this is all in BSL-3. obviously, and then measuring aspects of weight loss, viral load, initially in the lungs, where unfortunately the timing is a little unfortunate, I suppose, in that we've got a experiment, moving as we speak, and there is measurement of viral load in the brain in some infected mice that I was hoping would be ready for this grand rounds, but isn't. Well, anybody wants to know in the next week or so, I can give you an update. But we wanted to measure viral load initially in the lung. Then, for reasons I've mentioned before, some of the models that are used develop extremely high viral loads in the brain. We wanted to compare the gene replacement mice for that parameter, looking at tissues for histopathology. And then at later time points, start to measure aspects of the immune response, including serology for an antibody response and T-cell responses. And again, these are all on the B6 background, as Mike described. There's the wild type B6 mice, which we would not expect to get infected with this strain of the SARS-CoV-2, the K18 human H2 transgenic mice with that aberrant expression that Mike defined, and using two forms of the gene replacement mice, either H2 is on the X chromosome, so the gene replacement males or homozygous females. And these are some data I've shown before in the research forum. If we look at weight loss in these various strains, B6 mice, it's in a kind of gray, kind of purplish color in the back there. The B6 mice infected don't lose weight. The K18 transgenic mice rapidly lose weight and pretty much have to be euthanized by day 7 after infection. But neither of the gene replacement mice lost weight. Likewise, in a more recent experiment, I'm just kind of summarizing the genotypes here and showing individual lines. In a more recent experiment, again, the K18 mice lose weight. Some of these mice were taken at day 3 or 6, and so that's where the curves stop. The B6 mice, the controls don't lose weight. Gene replacement mice don't lose weight. One of them gained weight, which seems unlikely to be kind of typical, but they're not losing weight like the K18 mice. Oops. So this might mean that these mice weren't getting infected at all. So we have to be careful to see whether there's actually any changes in any ability of these mice, these gene replacement mice, to be infected by the virus. And that's where we started to look at viral load. My animations got messed up. This experiment, we were looking at the day 3 viral load. The B6 mice, the wild type mice, were infected, had negligible viral load. The K18 mice were infected. There's quite a bit of heterogeneity in actual fact in those mice. But reassuringly, the gene replacement mice, both the females and the males, heterozygous and homozygous, in terms of the copies of the gene replacement, were both infected in a very substantial way. This new experiment that's going on will assess viral load in the lung and the brains of infected H2 gene replacement in the K18 transgenic mice. And at the same time, where we have the tissues, we haven't submitted them yet, but they will be submitted as the comparative pathology shared resource for histopath. And if we look at serum antibodies, this is day 14 after infection. The K18 mice were all dead by this stage, had to be sacrificed, so we don't have numbers on those. But again, the wild type B6 mice were infected, basically undetectable serum response to the spike receptor binding domain. But pretty much all of the gene replacement mice showed a strong response. And this is just a negative and a positive control to make sure the ELISA was working well. So it would seem as if these mice, the gene replacement mice, do get infected with this SARS-CoV-2 isolate. They support viral replication in the lung at least, they don't die like the K18 transgenic mice from that infection, and at least for the antibody response, they mount a pretty robust serological response this time point. We're actively looking for the T cell response that we'd expect to be induced in these mice as well. So now that our initial premise was to generate this model. Obviously, thousands of other groups are also developing alternative ways to model SARS-CoV-2 infection in mouse models. So there are lots of other classic transgenic mice using human ACE2 cDNAs that have been used. I would say they pretty much all have the same kind of defects as Mike outlined. There's models in which people use intranasal infection with an adenovirus expressing human ACE2. That's acute, and that you can use pretty much any mouse strain you want and get this infection. The adenoviral vectors don't replicate and persist in mice, so this is a kind of one-shot deal. There's an expression of human ACE2 transiently in lung epithelial cells, and you have to kind of time your infection with SARS-CoV-2 to nail that. And of course, that doesn't model the physiological expression of ACE2 in other tissues aside from the lung. There have been mouse-adapted SARS-CoV-2 isolates, basically just doing sort of serial passage of SARS-CoV-2 in mice and looking for spontaneous mutants that show up that can now infect mouse tissues. Valuable resource, although you can have to wonder whether that what that is modeling and whether that's kind of accurate reflection of what natural SARS-CoV-2 does in humans. And now there are some natural variants. There's many more than this now. Obviously, Delta is on everybody's mind, and actually, I don't know off the top of my head whether Delta will infect via mouse ACE2. I'm feeling it doesn't, but could be wrong. All of these have limitations, as mentioned along the way, and often, ironically, the models are engineered to give fatal disease. So, for example, the mouse-adapted SARS-CoV-2, the reason they thought they were on the right track was that you keep going until you start having fatal disease infecting mice. So again, this is kind of focused on the fatal disease and not really developing models for less acute and lethal infection. And an important consideration that pretty much none of these cases is alternative splicing of human ACE2 for sure, it modeled. So as Mike had outlined, the natural expression of a gene is complex but not random. And there are, aside from the kind of steady state alternative splice forms, there can be instructions on how a gene is spliced and what proteins are produced, which will likely become important. So, for example, it's been reported before that interference may induce a truncated human ACE2 splice form that is not a receptor for SARS-CoV-2. We want that to be modeled in our animals, and that should be modeled in the gene replacement mice. That's the reference for that one. Potential uses of the human ACE2, and when we have them, ACE2 TMPRSS2 gene replacement mice, as we go forward, is to investigate how the expression of human ACE2 in extrapulmonary tissues affects disease after infection. And as Mike had mentioned, there sure is expression of ACE2 in the lung. Usually, people are, when you're trying to measure expression of ACE2, lung wouldn't be your top choice because the expression levels are pretty low and maybe not very uniform. Tissues like the kidney are incredibly good for a signal, and then heart, in some cases intestine. So most models aren't looking at that at all. They're focused, for obvious reasons, on the lung infection. But as we go further into this, and especially considering people who have been through infection with SARS-CoV-2 and survived, what are the effects of potential infection in other tissues aside from the lung? We want to also explore how previous microbial experience, such as using the dirty mouse model that we've developed and used in the department, how that affects infection, immune response, and disease progression. And how aging, more recent interest in how aging or the presence of senescent cells impacts SARS-CoV-2 induced pathology. So work that Sarah Hamilton and collaborators have led on the impact of senescent cells on kind of heightening vulnerability to infections. It'll be interesting to see how, in particular, the presence of senescent cells would regulate pathology after infection, either to maybe induce increased pathology in ACE2 gene replacement mice, or potentially to reduce the acute pathology in the K18 ACE2 mice. And obviously the platform here, once we have sort of completed the characterization of the ACE2 GR and the double gene replacement mice, we'd be able to introduce other gene knockouts or conventional transgenes if we need to modulate pathogenesis and see how that affects the response. There's also kind of related back to, I guess, to the first point, a need now to, well, I was going to say, as the pandemic winds down, which I'm not sure it is, but at some point there will be much more refocusing on how non-lethal infections are affecting the population. So models for MIS-C and MIS-A for antibody-dependent enhancement, for long haulers syndrome and so forth are going to be needed. And we're hoping that the gene replacement model will satisfy that need. Okay, that does it for me. Oh gosh, we've done it so early. That's good. So if there are questions that come up, feel free to ask Mike or I about them.
Steve and Mike, that was a phenomenal scientific accomplishment over the past year. So I give you tremendous credit for all of this work. And it's really amazing what you guys have achieved, you and the team, I should say. Do you know, maybe you don't know, but with the distribution of the animal lines out to various people, are people doing drug discovery on this to find, you would think they might, the pharma companies, because that would, we had some penicillin-like compound, it would be wonderful, or something like Tamivir or
Right. Yeah. So the way the Jackson distribution works, we don't really see who gets it and what they're doing with it. So it's made available. I know some people have written to me, and asked some details. So I know a little bit what's going on. Actually, one lab was really just interested in it in studying it for its ACE2 function. They were really interested in that. And so it's the human ACE2 gene, so that there are drugs that they're interested in with that. And so they're doing that. But right, we just don't know what's going on. I think this is actually the best model for looking at that. I mean, that's my bias, of course, but it's at the human level. So you're not working with overexpression. So if you wanted to block ACE2 receptor, it would be at the right levels and the right amount. And so if there was a inhibitor of ACE2 infection, that this would be really, you get accessibility of tissue issues that you can look at with the animal as well. So we just don't know. We put it out there, and I know it's being used, but and hopefully we get our description out before someone else does, but we don't know that either. So,
right. Yeah. Likewise for the TMPRSS2 gene replacement mice, when they're online, there's certainly have been inhibitors that block that protease and can reduce the best model in vitro, reduce the infection with SARS-CoV-2. Oh, and my video froze. if hopefully we'll be able to use the TMPRSS2 gene replacement mice to kind of get a kind of more accurate assessment of how those drugs will work in vivo, because they may well not work in the same way on the mouse version of the gene.
So could I jump in with a question here on the TMPRSS2?
Yeah.
Is the mouse TMPRSS2 functional in your system? Do you know?
It is functional, right? So we get infection, it processes. There are differences. There's significant differences between the proteins. So presumably, if you're working on an inhibitor, there might be differences with that. But
Sure.
when we do get functional infection, it is working.
Okay. Thank you. I didn't mean to interrupt, but I was curious.
Yeah, whether whether with the addition of human TMPRSS2, we would see, I don't know, a higher viral load in the context of the ACE2 gene replacement to be determined. Certainly, we can get a pretty good viral load just with the ACE2 gene replacement.
Plus, the inhibitors that Mike just mentioned in the human system will be much more, hopefully, clinically informative. So, I just want to echo Leo's. This is terrific. So, thank you.
Right. I mean, one of the advantages of working with the human gene is that the human products are human, right? So all the same reagents that you use for looking at a patient, you can use those same reagents in the animal.
Right. Absolutely.
I have a couple of questions. This is Brant Clark. The K18 mice, what's known about the expression of the ACE2 in the brain? I mean, it seems rather odd that a cytokeratin promoter would be expressing in brain.
Yeah.
Right. So I guess my point, I was trying to make the point was that's sort of just a starting point. That's part of the formula of what you get for expression, but really where you insert it has a huge impact on what expression you get. So that's probably inserted near some driver of neuronal expression, and you're just getting it just based on where it went in to the genome. And that's really standard with the transgenic. It's just somewhat random about the expression pattern that you get. And then if you're looking for a fatal disease, you infect your different lines and you find one that dies, that's what you got. But in this case, I think it probably was because they were getting expression in the brain.
And then sort of along the same line, what do you know about expression in the brain in your model? Yeah,
so we don't know a lot about that. So in here, I expect this, anything that we've looked at, it's really been the same as in humans. And in human, there is some brain, central nervous system expression, but it's really isolated cell types, and it's not easy to look at. And so we'll have to take a look at that. We just haven't done it yet. It might actually be easier to look at infection, see what tissues you can infect and do some sort of abnormal viral infection, and then look for viral signal, and then we can see functional expression that way. But that's all complicated infection outside of my realm of what we can do. Steve, do you know anything, if there's any interest in doing that?
Oh, sure. There's interest, and the experiments that are ongoing will, compare the viral load in lung versus brain of both the K18 and the gene replacement mice. We're expecting, although we may well be wrong, there would be minimal viral load in the brains of the infected gene replacement mice. Certainly predicting lower viral load than in the K18 ACE2 mice. But right, what is actually leading to that infection that gets to the CNS? Is it because there's a huge load in the lung transiently that then can efficiently and other tissues that can efficiently spread to the CNS, and then it's rampant, uncontrolled replication? Not really clear. So there's not been an awful lot of detail about the K18 ACE2 transgene expression in the brain. We've been looking for that, and it's been hard to spot. So I'm not sure that it's just completely uncontrolled expression in the CNS, but it'll be nice to be able to compare those two models.
So Steve or Mike Farrar, given the really high percentage of patients with severe COVID that also have either genetic defects in interferon or make antibodies to it, are you guys looking at what happens if you just add neutralizing antibodies to IFNAR in this model? Do they get more lethal disease?
Actually, we have it crossed now with that knockout, and so we've got some that are homozygous for loss of that receptor. So again, if there's some interest in doing these infections, we have mice now where we can look at that loss of interferon, and that is known that with the SARS-CoV-1 that makes a big difference. A lot of the SARS-CoV genes are really for neutralizing interferon response. So I would predict that will make a big difference, but we do have some mice now.
Yeah, although aside from the interferon receptor knockout, Mike raises a good point in that these neutralizing antibodies that have been defined in correlated with people who are very high risk of getting severe disease. It'll be valuable to be able to model that. Obviously, a little different from the knockout in that we can separate out transient exposure to the blocking antibodies and see how that affects things.
This is John Crossan. What's known, or I'm sure it's known, the difference in the amino acid makeup between the mouse ACE2 and the human ACE2, and does understanding this help to understand the increased infectivity, let's say, of the Delta variant or other variants?
You know anything about this, Steve?
Well, not the detail. I mean, certainly the basic difference in that the mouse ACE2 is does not bind to the receptor binding domain from the SARS-CoV-2 spike. That's been known for a long time. You can start introducing mutations into the mouse gene to try and kind of restore that functionality. Mm-hmm. The variants, though, that's a really interesting question. I'm not sure. Certainly, from looking at the variants that can now infect through mouse ACE2, they have been able to kind of pinpoint which residues are important for that capability. I think still the question is having a kind of side-by-side comparison, really, of being able to say, at a qualitative level, those variants can infect through mouse ACE2. Is it basically the same degree of efficiency as they would infect through human ACE2? That's you can mimic that in vitro, but these gene replacement mice would let us really do that comparison in vivo. So we haven't done that yet with the variants. We've been. Again, this has been a kind of patchwork collaborative process. So I think Mike and I have been pushing for some work with the variants that do infect through mouse ACE2. Is finding partners who will go to the considerable effort of growing up those variants and getting viral stock so that we can do the experiments. So we haven't got there yet.
I believe Maxim has a collection of variants, and so this would really be a nice model for directly comparing the biology of the variants, because you don't need to adapt them. You can use them as is, and just a direct comparison about the biology of the variants would be a nice system, because the genetics of the host could be exactly the same. Essentially, all of these mice are clones of one another, and so really the difference would be in the variant and not in the animal model.
Yeah, and we do have access to that collection of variants, but evidently growing them up to high viral titers sufficient for the kind of experiments we do is challenging. I think in the absence of additional directed funding, it'll be hard to rely on the kindness of strangers for all this.
This is Liam. So Mike, this is really a fantastic talk. I'm just wondering, since K18 mice, has such a high viral load and can, induce the cytokine storm, have people ever examined the brain from the K18 mice? Any pathology there?
I don't know. I haven't seen it, but have you seen the brain pathology in particular? I know they looked at viral load, but I don't know about pathology.
Yeah, that is one of the things where we have the tissues sitting in formalin right now, waiting to take over the comparative pathology core. These would be from day 3 and day 6 after infection. We managed to keep the K18 infected mice going until day 6 when they're right on the edge of needing to be euthanized. I think either of those time points when, at least from other people's work, there would be a high viral load in the brain. That'll be interesting to see. What the nature of that pathology is, and again, to compare it to the gene replacement mice. Don't have that answer, but it's certainly something we're interested in.
Hey, Mike and Steve, this is Andy Nelson. I have a much broader question. I'm going to leave SARS-CoV-2, sorry everybody else, and just ask about science. And I mean, I was fascinated by the stuff that really comes down to the question, Mike, that you showed with your data just on gene replacement in general, how nuanced it is to go from genomic sequence to a final protein product. For someone like me, and I think many others on the phone that, does rec and check with whole sorts of cell lines, in my case, breast cancer cell lines and things like that. I mean, should we, even with like cell line experiments, shorter term experiments, nothing quite as elegant as creating transgenic lines where you want to be much more, well, you want to be careful in every piece of science you do, but particularly when creating a transgenic, you want to make sure you don't screw that up. But I mean, should we be using gene replacement pretty much wholesale for every time that we want to pop in a different version of our favorite gene and study different effects of mutations or loss of function? Or
right. So I had a cartoon that made this look really pretty easy, but it's not. So this is a second example of a gene. So we replaced the tau gene and we replaced the ACE2 gene. So we've just got this up. It's really not at the stage where it's quick at all. So the standard approach for sort of careful biology would be to modify the genes that are there. And so when you're working with tissue culture, often you can work with the correct species. So if you're interested in human, you're using a human cell line. And so then it's sort of CRISPR modification of a human gene. That's really very accurate. Then you don't get an animal, but you get very good genetics with that. So if you can use the correct species, then you don't really need to do the whole gene replacement the same way you do when you're crossing species.
Yeah, I was just going to amplify the idea of CRISPR as being a game changer in terms of kind of accuracy and fidelity of these mutations in both human cell lines and in animal models. I mean, to make it even broader, something that my lab has been obsessing about recently is that we use a lot of what I call congenic mouse strains that are meant to be identical except for a single gene and a single amino acid in that single gene, and these were pretty much all made historically by breeding to inbred strains of mice, and then you backcross and backcross and backcross until you kind of lose the will to live. And you believe that you've now backcrossed it such that there's one gene, one exon, hopefully, that is different between these two mouse strains. When those have been looked at, probably no surprise, it's a massive chunk of genome that was sort of integressed from one inbred strain to another, often dozens of genes that came along for the ride. Jackson Labs now, and we got some of these mice in advance, made one of these congenic strains the right way with CRISPR, such that there is only a single amino acid difference between the original Jackson B6 strain and this one. And I suspect people will be flocking to that because finally you have something that's really clean. You don't have to worry about all the extra mud that comes along with what you'd like to think are carefully controlled experiments. I think different goals. Obviously, a gene replacement between species. You don't have many choices. You wouldn't want to have to introduce all those changes by CRISPR. But I think there's going to be lots of opportunities to modify things, and obviously we could modify the human ACE2 allele that we have in these mice, potentially with CRISPR, if we wanted to introduce a variant and see how that changes things.
Right, everybody. Thank you for questions today. We do have a 9 o'clock quality APCP meeting that we will log off for. Thank you, Dr. Jameson, and thank you, Dr. Koob.